First of all, I'd like to acknowledge the traditional owners of the land where I work and pay my respects to the elders past and present. So it's Pam's completion, PhD completion seminar today. It is a celebration rather than an examination process. But we like to hear any comments or suggestions, even at this stage, because Cindy can make use of those suggestions and comments in her thesis. She has listed her supervisors on the slide, and I'm Shamri Damage, I'm the advisory committee chair, and Kate, who is also on the advisory committee, is here. And Louise is the principal supervisor, and Richard and Martin are her co-supervisors. Cindy was attached to the Melbourne School of Population and Global Health, University of Melbourne, as a PhD student, but she was very much working between both institutes, the University of Melbourne and the School of Population and Global Health, and Florey Institute. Louise, yes.
Yeah.
Yeah. So that's where Anne-Louise is attached to now, although she has been in MCRI, as you know, for a very, very long time. So thank you for presenting your work, Cindy. And I'll hand over to you, and I'm sure you will outline all your achievements and what you have found in your thesis. Well done.
Thank you, Shamali, for that introduction. So thank you everyone for attending my completion seminar today. My thesis is entitled Early Origins of Childhood Neurodevelopmental and Emotional and Behavioral Disorders. And before we begin, I would like to acknowledge the traditional owners of the land in which we are meeting today, the Wurundjeri people of the Kulin Nation. I'd also like to pay my respects to elders both past and present, and extend the respect to other Indigenous Australians present today. So today I'll be going through 9 chapters of my thesis. That includes 3 published original work and 1 submitted this week. First, I'll go through chapters 1 and 2 that will summarize the introduction and literature review. So neurodevelopmental disorders, NDD, are characterized as impairments to the growth and development of the brain and central nervous system. These can impact a child's cognition, communication, behaviors, and motor skills. And for my PhD, I'll be focusing on 2 of the most common neurodevelopmental disorders, autism spectrum disorder, ASD, and attention deficit hyperactivity, ADHD. NDD accounts for 4 of 5 early childhood diagnoses in Australia, which is 1 in 100 children with ASD, or 1 in 20 children with ADHD. And trends show there is an expected increase over time. This makes NDD an epidemic public health priority. And both of these disorders are heterogeneous in nature, meaning they have both genetic and environmental impacts. I'll also be looking at emotional and behavioral disorders, also known as emotional and behavioral problems. I'll be using these interchangeably throughout my thesis. And these are broadly defined as emotional disturbances. If everyone could mute the cameras. So emotional behavioral disorders account for 1 in 5 children and adolescents worldwide. And there are 2 main domains of EBP. The first is internalizing behaviors, such as withdrawal and depression, and externalizing behaviors such as aggression disruption. EBP is associated with negative outcomes in children. These include poor education outcomes, high dropout rate, and comorbidity with mental health disorders. Mechanistically, EBP is poorly understood, but environmental factors have been shown to contribute to the etiology of EPD. In this study, I will refer to early life as prenatal, perinatal, and postnatal periods. And there are several routes that environmental exposures can be transferred to the mother. and the infants. These also include during pregnancy, placental transferal, and breastfeeding postnatally. There is currently no known cause of these disorders, and there are no effective treatment. Therefore, the World Health Organization has announced a global call to examine candidate determinants and programming mechanisms. Over the past 50 years, there is a concept called developmental origins of health and disease, DOHAD, that implicates that environmental exposures in early life contribute to fetal programming that maybe have lasting effects on the child's growth and development. And this is particularly true for early brain and behavioral development. This leads to my overarching hypothesis for my PhD, which is early life exposures, both genetic and environmental factors, impact key pathways such as oxidative stress, inflammation, and downstream pathways such as telomere shortening. And these underlying mechanisms contribute to increased cellular damage and consequently leads to the development of NDD and EPD in children. So first, I'll go through oxidative stress. So oxidative stress is a state of imbalance between free radicals such as reactive oxygen and nitrogen species production and antioxidant detoxification pathways. The redox equilibrium is important in preserving vital cellular function. And pregnancy is a state where adaptation and balance are disrupted and susceptible to oxidative stress vulnerability. The mother's adaptive mechanism also provides oxygen availability that allows for fetal growth and development. So two oxidative stress biomarkers I examine in this thesis. The first is 8-hydroxy-2-deoxyguanosine, 8-OHDG, which is a widely used urinary biomarker that is indicative of DNA oxidative damage. The other is 8-hydroxyguanine, which is 8-OH-GUA, is a base isomer of 8-OHDG and is reflective of RNA oxidative damage. And previous studies have shown that there are high levels of this biomarker in biofluids. To date, there are 2 studies that have reported on early life oxidative stress levels and neurodevelopmental and emotional behavioral outcomes in children. The first one on top here has shown that early life oxidative stress is associated with behavioral outcomes in children in school age. And the study below was not able to detect an association between prenatal oxidative stress and ASD outcomes in children. Next is inflammation. So inflammation is a common pathway of multiple diseases and infection. It is the natural defense mechanism of body tissues in response to injury, and inflammation can be harmful to the organism if the process occurs chronically. In the brain, inflammation impacts neuroplasticity and neurogenesis and has been shown to cause cellular damage to the central nervous system. In this thesis, I'll be examining 2 inflammatory biomarkers. The first one is high sensitivity C-reactive protein, HSCRP, which is reflective of an acute dynamic inflammatory response to exogenous stimuli such as infection. It is a widely used biomarker of inflammation and it has been shown to be indicative of acute inflammation. Glycoproteinocereals is the other biomarker, glyc-A, which is reflective of glycan complex and circulating acute phase protein levels. It is a newly described biomarker in both adults and children, and it has been shown to be reflective of both acute and chronic inflammation. So previous studies have shown that increased inflammation, mostly in HSCRP at age 10, is associated with neurodevelopmental and behavioral outcomes in school-age children, and there is less known on the glyc-A levels and its effects on children's neuro and behavioral outcomes. Next is telomere lengths. So telomeres are at the end of chromosomes that are essential for genomic integrity and are also known as cumulative oxidative stress biomarkers. Telomere lengths shorten with each DNA replication until it reaches a critical point and triggers cellular damage. Shorter telomere lengths have been associated with age-related neurodegeneration diseases in mainly adults. However, to date, there are a few studies that have looked at the association between shortened telomere length and childhood neurodevelopmental and emotional behavioral disorders. These studies here report a similar trajectory, that is, a shorter telomere length is associated with adverse neuro and emotional behavioral outcomes in children. So together, these significant gaps in the literature led to my PhD objectives, which aim to investigate the association between a diverse range of early life exposures, particularly environmental factors, oxidative stress in early life as a potential unifying mechanism, inflammation in early life as a key biological pathway, and telomere shortening in early life as a downstream pathway that may all subsequently impact the child's neurodevelopmental and emotional and behavioral outcome. So next I'll go through the general methods used to achieve these PhD objectives. So I used the Bio and Infant Study BIS as my study sample, which is based in Geelong, Victoria, Australia. BIS is a longitudinal birth population derived cohort with an antenatal sampling frame. It is made up of 1,074 mother-infant pairs, which include 10 pairs of twins. And the overall aim of this study is to investigate early life determinants and mechanistic pathways of a range of non-communicable diseases. This is unique due to its high array of data, including biospecimen measures. For my PhD, I used maternal urine for chapter 5 and maternal cord blood and infant blood at 12 months for chapters 6 and 7. Emotional and behavioral problems in children in this PhD were measured by the child behavior checklist at 2 years. And this is a 99-item standardized parent-reported questionnaire based on the rating of frequencies on a 3-point Likert scale. And evaluation is based on 3 types of scales: syndrome, composite, and oriented scales. All 3 scales were used throughout the thesis. At age 4, we used the strength and difficulties questionnaire. And this is a 25-item parent-reported questionnaire scored on a 3-point Likert scale. And evaluation is based on 5 subscales and a total difficulty score. So both the CBCL and SDQ are widely used and validated, widely used with validated psychometric properties and high test-retest reliability. Additionally, I also verified the ADHD diagnoses at age 4, which confirmed we had 9 cases. And these were done by retrieving initial medical records from pediatricians and psychologists. And there will be further discussion of these outcomes in subsequent chapters. So the first results chapter is on early life factors and ASD. So the thesis objective was to examine a large collection of early life factors and candidate factors that may influence oxidative stress that are associated with childhood neurodevelopmental outcomes. So this objective was addressed in a paper, first author paper entitled Early Life Environmental Factors Associated with Autism Spectrum Disorder in Children at Age 2, a Birth Cohort Study. This paper was published in Autism, the International Journal of Research and Practice, in the Wide Perspective for Autism Research in January 2022. The following QR codes will be they're available if you'd like to have a read of the papers. So just a brief recap on the background. So previous studies have shown that prenatal, perinatal, and postnatal environmental factors are associated with increased risks of ASD in children. And these same factors have also been linked to oxidative stress. This led to the study aims, which were to examine the collection of early life environmental factors, particularly those known to influence oxidative stress and ADHD symptoms in children at age 2 years. To investigate the prospective associations between early life environmental factors and ADHD in a population in Australia. And lastly, to assess the predictive performance of ADHD symptoms at age 2 on later ADHD diagnoses at age 4. For the methods, we used the Barwin-Eaton study as our study sample. The exposure measures, comprehensive questionnaires, clinical and biological data was collected across different social and environmental domains. These include prenatal, perinatal, and postnatal factors. In total, there were 111 factors that were examined. For the outcomes, we used the child behavior checklist, autism spectrum problems, both raw and T-score with a cutoff, a borderline to clinical cutoff, and the doctor-diagnosed ADHD cases. For statistical analyses, separate linear robust regression models adjusted for child sex and age at the time of behavioral assessment was examined. So first, the results. Here you can see that there are 38 key early life factors and ADHD outcomes that were correlated in the study. Lines inside the circle represent inter-class correlations, while lines outside here represent intra-class correlations. The red lines indicate positive correlation, and the blue lines represent negative correlations. The following factors were associated with increased risk of ADHD symptoms at age 2. For demographic and household composition factors, lower household income, lower maternal age, greater residential socioeconomic disadvantage, increased birth order, and lone parental status. One sec. Sorry. And the next is prenatal factors, including increased gestational weight gain, increased maternal stress and depression, and increased tobacco smoking frequency, and increased alcohol consumption. Perinatal factors included an air pollutant, particulate matter 2.5, and postnatal factors included increased maternal stress and depression, increased child secondhand smoke exposure, and increased home heaters that burn oil, kerosene, and diesel, and last but not least, increased exposure to mold and dampness. So we also examined whether CBCL's autism spectrum problems at age 2 was predictive of later ADHD diagnosis at age 4. And we found that the area under the curve was 93%. So this leads to my take-home messages. That is, a diverse range of early life factors are associated with increased ADHD symptoms at age 2. Most of these factors are modifiable. These include environmental air pollutant exposures, and early ADHD symptoms at age 2 are predictive of ADHD diagnosis at age 4. Next results chapter is on oxidative stress and emotional and behavioral problems. Okay. So the thesis objectives is to investigate a potential unifying biological mechanism, oxidative stress in early life, and to examine the associations with subsequent childhood neurodevelopmental disorders and emotional behavioral problem outcomes, and to examine the extent that early life factors are mediated by early oxidative stress to influence these outcomes in the children. So these objectives were addressed in a first author manuscript entitled Higher Maternal Oxidative Stress Biomarkers During Pregnancy partially mediate the effects of social and lifestyle factors on emotional and behavioral problems in early childhood. This manuscript was submitted this week to the Brain Behavior and Immunity Journal. So a brief recap on the background. So prenatal, perinatal, social and environmental factors have been associated with early oxidative stress. Oxidative stress has also been implicated as a key pathway in fetal programming. These prenatal and perinatal social factors have also been associated with childhood EBP in mostly school-age children. And there's a literature gap in the interplay between these three components, which led to the study aim, which is to investigate the prospective associations between early oxidative stress and emotional and behavioral problems in early childhood. And the second study aim is to investigate the prospective associations between early environmental factors and childhood EBP at ages 2 and 4 years. And we then evaluated the extent to which these effects of the early life factors on EBP are mediated by higher maternal oxidative stress in pregnancy. So for the methods, we used the Barwon infant study as our study sample. We used maternal urine at 36 weeks to measure two oxidative stress biomarkers. The first one is 8-OH-GUA and the other is 8-OH-DG. And these were measured by liquid chromatography mass spectroscopy, which I also assisted with the pre-processing of these data. The outcome measures, we used the child behavior checklist at age 2, total problems T-scores, and the strength and difficulties questionnaire at 4 years using the total difficulty T-scores. So I'll be calling these total EBP problems hereafter. For covariates, we examined determinants of EBP that have previously been described by other authors within the study. And these factors have been collected by questionnaires and clinical examinations. For statistical analyses, separate multivariable linear robust regression models adjusted for potential confounds and disease determinants. And we also examined mediation analyses. So the main results of this paper is that we found higher maternal 8-OH-GUA levels during pregnancy was associated with total EBP problems at age 4. Similarly, this association was also shown for total EBP at age 2, but the effects were less marked. We found that both maternal perceived stress and air freshener use during pregnancy was associated with childhood total EBP problems only. We also found that tobacco smoking and secondhand smoke exposure during pregnancy was independently associated with maternal 8-OH-GUA during pregnancy and total EBP in childhood, which is consistent with the literature. The following factors were independently associated with maternal 8-OH-GUA and the total EBP problems, EBP outcomes in childhood. And so therefore we examined mediation analyses. So the following factors: maternal education, residential socioeconomic disadvantage, maternal prenatal prescription medication, maternal prenatal tobacco smoking, and maternal prenatal secondhand smoke exposure were all partly mediated by higher maternal 8-OH-GUA during pregnancy. And so these factors were not considered in the fully adjusted model, as they may be on the oxidative stress causal pathway. So take-home messages for this paper is that higher maternal oxidative RNA damage during pregnancy is associated with greater EBP in early childhood. This is particularly true for emotional related problems such as depression and anxiety. The associations between several social and prenatal lifestyle risk factors, such as prenatal smoking on EBP in childhood, is partly mediated by higher maternal prenatal oxidative stress levels. So the next chapter is on inflammation, emotional and behavioral problems. So similarly to chapter 5, the thesis objectives are now replaced with inflammation. And so these objectives were addressed in a first author paper entitled "Infant Inflammation Predicts Childhood Emotional and Behavioral Problems and Partially Mediates Social Economic Disadvantage." And this was published in the Brain Behavior and Immunity Journal in May this year. So a brief recap on the background. Prenatal and perinatal social and environmental factors have been associated with early inflammation. And these prenatal and perinatal social and environmental factors have also been linked to EBP in mostly school-age children. However, there is a gap on the interplay between these three components in younger children, which led to the study aim, which was to investigate whether inflammatory biomarkers GlycA and hsCRP in cord blood and 12 months are associated with emotional and behavioral problems, internalizing and externalizing problems in early childhood. To investigate, the second study aim was to investigate whether early environmental factors influence inflammatory biomarkers GlycA and hsCRP in infancy, and to impact EBP in early childhood. So again, we used the bioinfo study as a study sample. We used cord blood at birth and whole blood at 12 months. And GlycA levels were measured using the nuclear magnetic resonance method, while hsCRP measures were measured using the enzyme-linked immunosorbent assay ELISA's method, which I also contributed to in the lab work. For outcome measures, we used the child behavior checklist at age 2, both internalizing and externalizing problems at T-scores. For the covariates, we used the determinants of EBP that has previously been examined in our cohort study. And these factors were collected by questionnaires and clinical examinations. For statistical analyses, separate multivariable robust linear regression models adjusted for potential confounders and disease determinants. Multivariable robust linear regression models with mutual adjustments for 2 of the repeated longitudinal measures within the same model, and mediation analyses. So the main finding of this paper was that higher GlycA at birth was associated with internalizing problems at age 2. And when we accounted for the baseline measure GlycA at birth within the same model with GlycA at 12 months, we found that GlycA at birth was the more important time point. Therefore, we focused on this hereafter. So factors that were independently associated with GlycA at birth and internalizing problems at age 2 were further examined by mediation analysis. Here, lower household income, which is a marker of social economic disadvantage, had a mediation effect and was partially mediated by GlycA at birth by 6%. Other factors that also had an indirect effect or mediation effect included multiparity mediated by 12%, and greater number of older siblings aged 2 to 6 years mediated by 13%. It's important to note that multiparity and a greater number of older siblings were correlated. So together, these factors were not considered in the fully adjusted model as they potentially may be exerting an effect on the inflammation causal pathway. So the take-home message for this paper was that higher glycane levels in cord blood at birth is associated with greater EBP at age 2. This is particularly true for internalizing problems in children. And the effect of social economic disadvantage markers on EBP in childhood is partly mediated by high inflammation levels at birth. The next results chapter and the final one is on telomere shortening and ADHD. So the thesis objectives were similar to chapters 5 and 6. However, now we've replaced it with telomere shortening. So these thesis objectives were addressed in a first author paper entitled "Shortened Infant Telomere Length is Associated with Attention Deficit Hyperactivity Disorder Symptoms in Children at age 2 years, a birth cohort study." And this was published in the International Journal of Molecular Sciences in a special issue called Advances in Neurodevelopmental Disorders Research. And it was published in April this year. So a brief recap on the background again. Prenatal and perinatal social and environmental factors have been associated to shorten telomere length. Both previous family and twin studies have shown that telomere length is highly heritable. And these prenatal and perinatal social and environmental factors have also been linked to ADHD in mostly school-age children and adolescents. Other previous studies have also shown that ADHD is between 75 to 90% highly influenced by genetics. Therefore, there is a gap on the interplay between these three components, which were examined in the study. The study aim was to investigate whether telomere length at birth at 12 months were associated with ADHD symptoms in children at age 2 years. And we used the Barwin infant study as a study sample. Cord blood at birth and whole blood at 12 months were used for DNA extraction, and telomere length measures were measured by the quantitative polymerase chain reaction, which was completed by another PhD student. The units for telomere lengths is the TS ratio, which is telomeric genomic DNA over beta globin single copy gene. Which is reflective of a genetic component. Outcome measures is a child behavior checklist, which we used the attention problems and the DSM-5 attention deficit hyperactivity problems at age 2. For covariates, we used the determinants of EBP that have previously been examined in this cohort, and these factors were collected by questionnaires and clinical examinations. For statistical analysis, separate multivariable robust linear regression models adjusted for potential confounders and disease determinants, multivariable robust linear regression models with mutual adjustments for 2 repeated measures within the same model, and mediation analysis. So the main finding of this result of this paper was that shortened telomere length at 12 months was associated with greater attention problems and attention deficit hyperactivity problems in children at age 2. And when these 2 time points, telomere birth and telomere at 12 months, were mutually adjusted within the same model, we found that telomere at 12 months was the more important time point. Therefore, we focused on this hereafter. Here, we examined an extensive list of factors that were associated with telomere at 12 months and the attention ADHD symptoms. But I just wanted to highlight that child secondhand smoke exposure at 1 month was independently associated with telomere length at 12 months and the ADHD outcomes at age 2. However, we were not able to detect a mediation effect. Therefore, this factor was considered within the fully adjusted model. So the take-home messages is that shorter telomere length at 12 months is associated with increased ADHD symptoms at age 2. A diverse range of factors is associated with shortened telomere length or ADHD symptoms only. And child secondhand smoke exposure at 12 months was independently associated with shortened telomere length in infancy and ADHD symptoms. So lastly, I'd like to go over chapters 8 and 9 on the general discussion and conclusions. So the following early life factors were associated with neurodevelopmental and emotional behavioral problems in children at ages 2 and 4 years. I've categorized them into 4 domains. The first one is demographic household composition, prenatal, perinatal, and postnatal factors. So these factors were shown to be important to neuro and emotional behavioral problems across the 4 results chapters. And you can see here in red, these 2 factors, lower household income and increased residential social economic disadvantage, were consistently associated across all 4 chapters. Here, by looking at the list, I'd like to emphasize on the prenatal stage that may be a key a vulnerable, sensitive stage that may impact early brain development. So the study enhanced knowledge of the etiology of neurodevelopmental and emotional behavioral disorders. As shown previously, several early life factors come together to impact these outcomes. I also found that higher oxidative stress during pregnancy, particularly 8-OH-GUA levels, which is reflective of oxidative RNA damage, is associated with EBP in early childhood. Increased inflammatory biomarker glyc-A at birth is associated with greater internalizing problems at age 2. And shortened telomere length at 12 months is associated with greater ADHD symptoms at age 2. So previously, my overarching hypothesis was that all of these biological pathways may be linked in the children. However, I found that these biological pathways were not correlated, and therefore they may be acting independently to contribute to the development of neuro and emotional behavioral problems in young children. This leads to my implications and future directions. That is, categorized into 4 stages. The first one is preconception. So there should be more emphasis on addressing the socioeconomic gap as a public health priority. During the prenatal stage, advances in other determinants of childhood EBP and NDD. Prevention strategies to risk factors that may influence fetal programming, to target intervention to minimize inflammation levels, to understand the role of telomere shortening during pregnancy, to target interventions to maintain oxidative stress at optimal levels. In the perinatal stage, to target intervention to minimize inflammation levels at birth, and both during perinatal and postnatal stages, to understand the role of oxidative stress in infancy, and to further advance on the role of cumulative inflammation levels in infancy to optimize the child's healthy neurodevelopment and well-being. Lastly, I would like to emphasize that the prenatal stage may be an opportune time to intervene early. So these are my overall PhD outcomes in the past 3.5 years. This includes 4 first author papers, 3 published and 1 submitted. I was lucky to present at 8 conferences. I was awarded 5 scholarships and travel grants. I was also awarded the Murdoch Children's Research Institute MCRI's Rising Star Award in 2020. I've also been nominated as one of MCRI's top PhD candidates for a prestigious Australian award called CSL Florey Next Generation Award. I've enhanced skills in epidemiology and statistics. For example, I've learned to program in 2 statistical programs. And lastly, I've survived a PhD amidst a global pandemic. So lastly, I'd like to acknowledge my supervisors, Anne-Louise, Richard, and Martin, and for their ongoing support and guidance, and my advisory committee, Shamali and Kate, for their wisdom and deep compassion when challenges arose across the 3.5 years. I'm incredibly lucky to have 2 supportive teams, both the neuroepidemiology team and the molecular immunity team. Thank you to the participants and the investigator group. Without them, this PhD wouldn't be possible. As well as sharing my deepest gratitude towards Life Course for their financial support and training. This PhD would not have happened without them. To my external collaborators, it's been such a pleasure working along such great scholars. But lastly, but not least, I'd like to thank my family, both my ba and my mum, Mia, who is also here today, my partner, Sam, my friends, and the Scholar Diaries community for their constant report and believing in me when I found it hard to believe in myself. Thank you, guys.
Well done, Cindy. That's a fantastic presentation. As always, they were great slides and they were very clear, and you told us a great narrative. Congratulations on all your achievements, and also congratulations to your supervisors for supporting you to achieve such comprehensive outcomes. So it's open for questions and comments from the audience. You could either put your hand up or you could just talk if you have any questions. I don't see any hands yet, so I'll make a start. So Cindy, the last slide, you put up the last slide and you put the preconception period and as the first one, although you haven't looked at the preconception period, I guess whatever the social disadvantage at the time of the birth must be related to the preconception period as well. But could there be other factors in that preconception period, similar environmental factors that are influencing the offspring health outcomes that will pass through the generations? So I was interested to see that you just put the socioeconomic disadvantage.
Yeah, so you're correct, Shamli. So I placed the social factors before the prenatal birth stages because I believe that these would have been pre-occurring before the child is conceptualized. And I do think there are other factors within the preconception stage that we have not examined only because this is a birth study, which was, I guess, established when the mothers were during pregnancy stage, and so other factors may have not been measured that may potentially do have a transgenerational effect on these children. Yeah.
Thank you. One more question before I hand over to Kate. The telomere length and the ASD, you sort of outlined the causal pathways and the different arrows from one to the other. I didn't see common genetic causes there. Could there be common genetic factors related to both ASD and telomere length?
Yes, I think there would be a link between genetic factors and telomere lengths and ADHD. However, I didn't have such a large genetics component within my PhD. So I think this would be a really informative and very interesting, I guess, topic to look at in future studies, because both have high influences regarding to the genetics.
Thanks, Cindy. Over to you, Kate.
Thanks very much. Well done, Cindy. Just terrific to see your work and a beautiful presentation. So thank you and congratulations on such an achievement. Shamli stole my question. So I was going to ask you genetics too, because I just thought key factors, and I'm aware that Biz probably has that. So I agree with you. It's a really interesting next step. Yeah. My other question was just in regards to the environmental exposures that were related to ASD. So there were a whole host of things. I think there was exposure to herbicides, and I was particularly interested in air pollution. And you mentioned the SES ones were like the biggest factors throughout those periods. But in terms of the environmental factors, was there one that particularly stood out, or were they all just very small contributions to ASD?
I think so. Based on the key early life factors, there was 38 of them. The factors that were consistently associated with both ASD symptoms at age 2 and ASD diagnosis at age 4 were summarized on the slides. So those that I've included on the slides were consistently associated with both. However, out of those factors that I've listed, the most, I guess the one with the most strongest effect would be around the social economic disadvantage factors, so related to household income, residential social economic disadvantage, those factors, yeah.
Okay, thanks. Interesting.
Thank you.
Okay.
Does anyone else have any comments? If not, let me congratulate you again, Cindy. But before I do something that I'm supposed to do on behalf of Mark Jenkins, the director of the center, I would like to give an opportunity to Anne-Louise if she likes to say anything, Anne-Louise.
Yeah. No, yeah, I'd like to just add my congratulations. Well done, Cindy. You really grappled with some difficult issues in the PhD. It's one thing to describe the surface epidemiological association. It's another thing to try and work out underlying mechanisms. And you did that in several areas within your thesis, and the rest of the team enjoyed working with you on all these issues and seeing this progress so well, leading to 4 papers in the end. So all the best with your next chapters. And you really are quite skilled now too, going forward with various epidemiological and biostat concepts. So hopefully that'll stand you in good stead too. So well done. Yeah.
Thank you.
Richard and Martin, do you have anything to say?
I just say it's been a pleasure to be working with Cindy. She's been obviously my position is as a statistical advisor, so I've found her to be very willing to take on new aspects of learning, and nothing has seemed to completely faze her about the more technical challenges of what she's done. So I'm most impressed, and she should be very proud to reach where she has.
Thank you, Marty.
I don't see Richard. I thought I saw him before. That's okay. Cindy, can you unshare your screen? I want to share something.
Of course.
Yeah.
Yeah.
Can you see that? So this is something that our center does. Unfortunately, Mark can't be here today, and he usually presents a certificate of completion at the completion seminar. So on behalf of Mark, I would like to present the certificate that's on the screen. If we were under normal circumstances pre-COVID, we would have presented that to you physically, but it will be posted to you or in the email to you. So well done and congratulations. And I should also take the opportunity to congratulate your parents on your achievements. And I can see them taking lots of photos. They must be very proud and they must be very happy for you, Cindy. So well done.
Yeah. Thank you. Yeah, so thank you so much, Shamli. That meant a lot. I'm a first generation student. I'll be the first in my immediate family and the first in my whole ancestry history to have a PhD. So it's a phenomenal feeling.
Congratulations again and well done.
Thank you.
Thank you for the great presentation and congratulations to your supervisors again. Enjoy.
Thank you.
Have a
Thank
nice
you, everyone.
celebration.